Welcome to this explainer. Let's jump right in because we are looking at something absolutely incredible today. Sickle cell anemia isn't just any disease. It actually holds a deeply historic place in medical science. Believe it or not, it was the very first human disease to be understood completely at a molecular level. Today, we're going on a journey. We're going to start at the absolute microscopic level, analyzing a condition of stunning architectural precision, and then zoom all the way out to modern, definitive genetic cures. We're going to unpack the causes, the symptoms, and the groundbreaking science that is literally changing lives as we speak. Okay, let's dive into this. It's a pretty profound question to start with. When you think about a massive, devastating, multi-system disorder in the human body, you'd probably assume it has some incredibly complex, chaotic origin, right? But what if I told you that all of this physiological devastation starts with a tiny, practically invisible typo? Just one single molecular error written into the biological text of our DNA. Section 1. The microproblem, a single genetic typo. Let's put on our molecular detective hats for a second. At its core, sickle cell anemia is an autosomal recessive genetic disorder. That's a formal way of saying you inherit it, and it's caused by a highly specific point mutation on chromosome 11. Specifically, it happens right inside the HBB gene. Now, why on earth does this specific gene matter so much? Well, it acts as the exact instruction manual for the beta-globin subunit of hemoglobin, that absolutely vital protein in your blood that carries oxygen everywhere. The entire foundation of this disease rests on one incredibly precise nucleotide substitution inside that manual. And this brilliantly illustrates the exact problem. Notice the stark contrast in the biochemistry. Normally, we have the code GAG, which produces an amino acid called glutamic acid. This acid is hydrophilic, meaning it absolutely loves water, and it sits happily in the watery environment of your blood. But then we have the mutant code. A single thymine to adenine base swap changes that code to GTG. Just that tiny swap replaces glutamic acid with valine at position 6 of the beta-globin chain. And here is the absolutely critical detail. Valine is hydrophobic. It literally fears water. So instead of a water-loving component, you suddenly have a water-fearing one embedded right into the hemoglobin structure. So how does that actually play out? Let's walk through the precise biochemical cascade that happens when this altered hemoglobin goes to work. Step 1. The hemoglobin drops off its oxygen in the body. It deoxygenates. Step 2. Because that new valine amino acid is terrified of the surrounding water, it desperately seeks out and sticks to neighboring globin chains just to hide from the fluid. And step 3. This microscopic stickiness forces the hemoglobin molecules to stack and polymerize into these long, rigid, fibrous cables inside the cell. It's a complete, fundamental distortion of the cell's internal architecture, and it's all triggered by that one little fear of water. Section 2. The macro fallout, the devastating physical toll. To put this into perspective, the numbers here tell a really tragic story of structural failure. Normally, a red blood cell is this flexible, biconcave disc. It glides effortlessly through your tiniest blood vessels and lives for a robust, healthy 100 days. But when those rigid hemoglobin cables form, they physically distort the cell into a stiff, crescent, or sickle shape. These sickled cells are incredibly fragile. They're super prone to premature rupture, a process called hemolysis, and they only survive for a mere 10 to 20 days. The body simply cannot keep up with that kind of rapid destruction. And unfortunately, this brings us to the severe systemic failures these patients have to endure. Because these sickled cells are so rigid and sticky, they snag on the walls of narrow capillaries. They create these microscopic log jams that completely block blood flow. This causes vaso-occlusive crises, which basically deprive downstream tissues of oxygen and trigger excruciating, unpredictable pain, usually in the bones, chest, and abdomen. Then there's acute chest syndrome, a leading cause of mortality, where these blockages happen right in the lungs, causing hypoxia that mimics severe pneumonia. The spleen gets rapidly overloaded and repeatedly infarcted by these clots too, leading to functional asplenigia, which basically means the spleen just stops working, leaving the patient highly vulnerable to bacterial infections. Over time, these repeated microinfarctions damage the kidneys, the liver, the brain, and significantly increase the risk of strokes. It is a massive, system-wide toll. Section 3. The bone marrow bottleneck, the historic cure. Let's move to and see how this builds into the quest for a cure. Historically, the only clinically validated cure has been an allogenic bone marrow transplant. The concept is kind of brutal, but effective. You destroy the patient's defective bone marrow using chemotherapy, and you replace it with healthy stem cells from a matched sibling donor.
If it works, the body completely resets its red blood cell manufacturing line and starts producing healthy, normal hemoglobin.
But there's an agonizing catch, and it comes down to the numbers. Less than 20% of patients actually have a matching unaffected sibling donor. That leaves a staggering 80% of patients completely without a match. And for that 80%, attempting a transplant with a mismatched donor brings a severe risk of graft-versus-host disease, where the donated cells actually turn around and attack the patient's body. For decades, this massive donor bottleneck left the vast majority of patients entirely without a definitive cure.
Section 4. The microsolution. The genomic revolution.
But what if you didn't need an outside donor at all? So the crucial point is, modern hematology has achieved a truly transformative milestone with autologous gene therapies. Autologous simply means the cells come from the patients themselves. By taking the patient's own stem cells, modifying them in a lab to fix or bypass that genetic error, and giving them back, we eliminate the donor bottleneck entirely. And because it's their own tissue, that terrifying risk of graft-versus-host disease, it is completely bypassed.
Now, what's really interesting about these two regulatory-approved strategies is how brilliantly different they are from one another. Let's break them down. Strategy 1 is fetal hemoglobin reinduction. It uses CRISPR-Cas9 gene editing to disrupt a specific gene enhancer called BCL11A. Think of BCL11A as a molecular switch that normally turns off the production of fetal hemoglobin after we are born. By breaking that switch, fetal hemoglobin turns back on. This healthy fetal hemoglobin floods the cell, diluting the defective sickle hemoglobin and entirely preventing those rigid cables from polymerizing. The second strategy is gene addition. Here, scientists use a lentiviral vector to deliver a brand new, highly engineered anti-sickling beta-globin gene directly into the cells. It acts like a direct instruction manual telling the cells to manufacture a variant of normal adult hemoglobin that actively blocks the sickling mechanics. Two completely different scientific paths, but both achieving clinical cure status.
And the results of these genetic therapies? They are nothing short of triumphant. Data from pivotal clinical trials show that over 90% of patients treated with these specific therapies remained completely free of those severe, excruciating vaso-occlusive crises for years after treatment. Over 90%. They are quite literally getting their lives back, finally free from the shadow of those microscopic logjams.
As the source material so perfectly summarizes, recent breakthroughs in peer-reviewed literature have shifted the paradigm from chronic management to definitive genetic cures. We have literally witnessed the evolution of a disease that for decades meant a lifetime of pain and palliative care, transforming into a condition that human ingenuity can now definitively cure at the genetic level. This whole journey, from understanding a single thymine to adenine swap all the way to CRISPR-Cas9 therapies, is just awe-inspiring. But it leaves us with a fascinating, provocative thought to end on. If we now have the technology to reach into the human genome, find a single genetic typo, and successfully rewrite it to cure sickle cell anemia, what other complex, devastating diseases out there are just waiting for a molecular proofreader? The genomic revolution is really just getting started, and the possibilities are absolutely limitless. Thanks for joining me on this explainer, and keep questioning what's possible.